Good morning. Today we are going to cover topic 12 and we're going to do a recap of 12.1, which is plant nutrition first. So the learning objective for plant nutrition is that you need to define photosynthesis as the process by which plants manufacture food using carbon dioxide and water in the presence of sunlight and chlorophyll. The second learning objective is to state the word equation for photosynthesis, but I'm also going to teach you guys on how to write down the symbol equation as well. For now, we need to check your prior knowledge first. So for this topic, you need to state the definition of photosynthesis. You need to be able to give in a reason why it is important for plants, and to also explain how plants manufacture carbohydrates. You also need to be able to state all the raw materials and also the products of photosynthesis actually. The photosynthesis, how do we define it? Photosynthesis is the process by which plants make glucose, aka their food, from raw materials like carbon dioxide and water, using the conditions light energy from the sun, and oxygen becomes a byproduct. What does raw material mean? Raw material means that they are the things, the compounds, or the elements that are used up to make glucose, which is the food. The conditions here is that the plants need light energy from the sun, and to help them gain that light energy, the plants actually have chloroplast, and chloroplast contains chlorophyll, which is the pigment that absorbs the sun energy. Why is photosynthesis important for plants? Plants need photosynthesis, this process, to produce food for them. And also it's important for other living things, whereby this food being produced will be used for other organisms that actually feed on the plants. Manufacturing means to make and assemble carbohydrates. Carbohydrates is a group of nutrients, and there are different types. There's really simple carbohydrates, there are complex carbohydrates as well. So these plants, they actually make simple carbohydrates, which is glucose, but that can actually be converted into a complex carb, for example, starch. So how do they manufacture, how do they make carbohydrates? They use small molecules that are absorbed. And the small molecules here we're talking about is carbon dioxide and also water molecules. And they synthesize, so they put together all of these small molecules to make bigger molecules, which is the glucose molecules. And eventually it is stored as starch, which is a complex carb. Energy used for this process comes from light energy from the sun. So this is the equation for photosynthesis. And you can see here we have one small molecule here, which is CO2 plus H2O. And after absorbing light energy, we get this big molecule right here, which is glucose. And then you plus right here, oxygen gas is released, another smaller size molecule. Glucose, you can see, is actually much more bigger, and there's a lot of lines right here, and these are actually called the bonds. So there's many bonds here, and that means that there's many energy stored in there. This is a learning objective that you'll get in year 9 biology, which is to state the equation in words or symbols for photosynthesis. So the word equation for photosynthesis is pretty simple, and this is what you need to know for your exams for year 8. So you have the same process going on there, that's photosynthesis. So the word equation is just, you need to write down here for glucose, just write down glucose, not chemical energy. You write down light energy on top of the arrow. And this is the word equation. It's just writing down the words carbon dioxide plus water arrow and then light energy and chlorophyll at the bottom. And then you write down glucose plus oxygen. As for the symbol equation here, you write down CO2, H2O, C6H12O6, and O2, and make sure to write down the words light energy and chlorophyll above and below the arrow. So this is a little bit special in the fact that the symbol equation needs to be balanced. So if you look over here, why is it C6H12O6? It's because this glucose molecule is made up of 6 carbon, 1, 2, 3, 4, 5, 6, made out of 12 hydrogen, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 6 oxygen atoms, 1, 2, 3, 4, 5, 6. That's why it's called C6H12O6. And plus O2. And if we want to balance this out, that's why we put 6 in front of carbon dioxide, water, and also oxygen gas. So I asked you guys to draw a flower before the holidays. And the fact that this is pretty important for animal nutrition as well. So with plant nutrition, we know that plants, they make glucose, and that's their main source of food. So add the leaves. But what happens is that after pollination is when the pollen grain will fall onto the stigma, and then it's going to make its way into the ovule through the ovary first. And then once the male and female gametes, they fuse, they will actually turn into a new young plant. So this flower right here, once pollination occurs, this ovary, which is the covering of the ovule, it will become the fruit, fruity part right here, the flesh of the fruit. Whereas the ovary, the ovule, over here, the little dots right there will become seeds. So this fruit, this tomato fruit, actually comes from a flower. This is showing you guys the process. So we have a flower here, and after pollination, the petals actually fall off, and then what's left is a sepal and also the ovule inside and the ovary at the outside, and it becomes bigger and bigger. How does it become bigger and bigger, and how does it actually grow? Is that the glucose from the leaves actually supply it with a lot of nutrients. It's so that the seeds can use up that nutrients to grow into a new plant in the future. Now we're going to look into topic 12.2, animal nutrition. In this unit, you will learn about the primary sources of food, how food is a source of energy for life, how to test for the major components of the human diet. And for this topic, we need to have a lot of prior knowledge. The recommended prior knowledge is number 1, you need to know the existence of chemical elements, and we actually covered this in year 7. You need to know that chemical energy is contained within larger organic molecules, but you don't know this very well yet because you're only going to learn about this in chemistry in O levels. You also need to know a lot about enzymes and enzyme action, and we covered this just a little bit from before. We've talked about it. But also, mainly, you also need to know that animal nutrition is related to the simple understanding of the circulatory system, which was covered before the holidays. So let's do a recap of your elements and compounds. So what is the word matter, and how does it relate it to molecules, atoms, and particles first? Matter, from big to small, we're moving in from matter first. Matter is anything that can take up space. It is made up of subatomic particles like atoms and molecules. Molecule is when you have 2 or more atoms joined together. A very famous example of a molecule is water. So you can see that oxygen here, an atom of oxygen here is connected or bound to 2 hydrogen particles right there. What's an atom then? atom is made up of smaller particles that are called proton, neutron, and electron. And you can see right here, this in the middle part here is called a nucleus, and surrounding the nucleus is electron. Inside the nucleus, you can find proton and neutron. And what's a particle? Particle is any tiny bits of matter. Sometimes atoms, they are called particles, but we usually refer them to as atoms for one of them. And they can have different sizes. So protons, neutrons, atoms, and even molecules are considered as particles. So if it's like one part of something big or a group of something, they are just called particles. But an atom is specific in terms of the type of atom, which we'll talk about after this. The molecules can either be elements or compounds. A molecule can be an element kind of molecule, and this means that you have two or more of the same type of atom chemically joined together. A molecule can also be a compound, which is when you have two or more different types of atoms chemically joined together. An example of a compound is water, and an example of an element is oxygen. You can see here for the oxygen gas that there is two of these two atoms of oxygen joined together. But for the molecule that is a compound, which is water molecule, there's two or more different types of atoms chemically joined together. The two types of atom here is two atoms of hydrogen, and the other type is this oxygen right here. Only one molecule, only one atom of oxygen is connected to two atoms of hydrogen. Molecules are found in living things as well, and it's not just water, and it's not just in the air, but it is in you and me. And these molecules that can be found in living things is called a biological molecule. A biological molecule is a molecule that is present in living organisms. So all of these things, even plants, humans, and insects, there are molecules in all of us. So how are molecules formed? Molecules are made by forming bonds between atoms. There are different types of bonds. There's covalent bond, ionic bond, and hydrogen bond. And the main one for living organisms is actually the covalent bond, but there are also other of these important bonds as well. You're going to learn about the bonds between atoms in O levels in the future. So actually, when atoms join up together, they actually store energy in that bond. So the energy is stored in the covalent bonds holding atoms together in the form of molecules. And this type of energy, where does the word energy here? The type of energy is often called chemical energy. So right here, this is a diagram of an amino acid. An amino acid is actually a subunit of protein. So the protein that you can find in chicken and in beef and in fish, they are all made up of these tiny bits of amino acids joined up together. And one of these circles right here, one amino acid looks like this. It's made up of even more like bonds right there. So right here you have a bond, and right there you have another bond, and you actually have other bonds as well. And if you imagine that's just one amino acid, and imagine all of the ones right on this beaded structure right here, that means they have like a lot more bonds. That means there's more chemical energy being stored in the protein molecule. All right, the circulatory system, how does this relate to everything we're talking about? The circulatory system is a network consisting of blood vessels, and the heart. This is a network that supplies tissues, so parts of your body, with nutrients, oxygen, hormones, and removal of waste. So the circulatory system, what it does is that it actually transports the nutrients that you need, and that you actually, after you digest the food, you get these nutrients to all parts of the body, because the tissues need it for other purposes, like energy, for example, forming new tissues, for example, to synthesize stuff. So not only do plants can synthesize things, but you can as well. So now we're going to look into the learning objective. So 12.2.1, you need it to state that plants are the primary source of food. And you also need to state that food is a source of energy for living things. You also need to describe the dietary importance of protein, carbohydrates, fats, vitamins, mineral salts, water, and fiber, aka roughage. Why are nutrients important? Plants are the living organisms, and what they do is that they produce their own food by the process that we talked about earlier, which is photosynthesis. This makes them an important food source for living things. So animals, like humans, we need food to, number one, to repair worn out tissues. So tissues like the white blood cells and cells like the red blood cells, they actually wear out, and within a few months you're going to get new red blood cells because you use them quite a lot and they get pretty worn out. Worn out means that they won't function as well as they did before, and especially the white blood cells. White blood cells go through a lot. They actually fight a lot of bacteria and viruses in your immune system, so they get really worn out. So their quality isn't as good, so you need to make new ones, and you need to have nutrients to make new ones. The second important function of food, aka the nutrients, is to maintain a healthy body. Third is to grow new cells and new tissues. And the fourth one is to provide energy for daily activities, for example, walking. What does the word nutrient mean? Nutrients are the chemical substances that you can find in the food that you eat. So for example, we have an apple, but it contains all of these things. So it doesn't just contain vitamin and minerals, but it also contains a lot of carbohydrate, and the most, which is 20%. Dietary fiber, that's your roughage. And actually your apple contains a bit of protein as well, around 1 gram. So the nutrients that can be found in the apple, you can find minerals, vitamins, carbs, fiber, and even protein. So they're all actually chemical substances, like the chemical elements that you can find in your body. They are also molecules as well. Carbohydrate molecules are biological molecules that are made up of these elements. So we talked about elements just now, and you can find that in your periodic table. So the word carbohydrate comes from the meaning carbon plus water. And hydrate part of carbohydrate, it actually comes from the fact that hydrogen and oxygen atoms are present in the ratio of 2:1, just like in water. So these are the elements that you can find in carbohydrate. CHO, CHO. There are different types of carbohydrates. So I talked about it, like you have complex carbs and simple carbs. This is an example of a simple carb called monosaccharides. You have disaccharides, which is also a simple carb, and you have your complex carb, which is polysaccharides. So if you look at the monosaccharide, it's actually a very simple carb. So this is like glucose, the ones that the plants make during photosynthesis. So it's just 1 unit of carbohydrate. Fructose and galactose. Fructose is a type of monosaccharide you can find in fruits, and galactose in milk. Disaccharide is when you have two molecules of carbohydrate being joined together, and you can find maltose in a lot of fruits as well. And you also have sucrose, which is found in plants. This is the type of sugar that you can find in your tea. And you also have lactose, which is a type of sugar found in milk. And polysaccharides, if you look over there, so it comes from like small, so just 1 unit, to two units being joined together to like many units like joined up together. This is why we call starch a complex carb. But the main things you need to know for your 8 is the word glucose, so that carbohydrate, and also the complex carb, which is starch. Starch, you can find them a lot in potatoes. Glycogen is a polysaccharide, a complex carb that is found mainly in your liver and your muscles. And cellulose is a complex carb that you can find in mainly all plants, because the cell wall of plants are actually made up of cellulose. Okay, now let's look at fat. So this is another main component of the human diet. Fat molecules are biological molecules that are made up of the elements carbon, hydrogen, and oxygen. So that's actually the same as the chemical elements found in carbohydrates. But what is the difference? Unlike carbohydrates, fats contain less oxygen in proportion to hydrogen. So let's have a look how it looks like. So here is your glucose I can find in honey, and this is cisterine, which is beef fat. You can see over here that the ratio is 1:2 between hydrogen and oxygen, like in water. But for for fat, you can see that there is much higher hydrogen in ratio to the oxygen. So all biological molecules always contain carbon and hydrogen. Protein molecules contain these elements, CHO and also N and S. N here stands for nitrogen and also sulfur, whereas carbohydrates and fat contain C, H, and O. Now we're moving on to the 7 nutrition groups. So how do my previous year 9s remember this? They taught me this mnemonics, which is Jegu Fatima plays the violin for my wedding. C is carbohydrate, F for fats, P for protein. Then you have V for the violin is vitamins, F for fiber, M for minerals, and W for water. These are actually how your molecules look like. So carbohydrate, you can see that there are little atoms right there joined up together, and that's glucose. And when you get more of those glucose molecules joined up together, you make a complex carb like starch. Fat is also made up of molecules, protein, vitamins, fiber, and even water. Now let's talk about the sources. So sources means like, where do they come from? Like, where can you find all of these nutrients, and why are they important for your body? Firstly, we have carbohydrate. Carbohydrate is a molecule. Carbohydrate molecules, they contain a lot of energy, which is released in the cells, and they are broken down during the process cellular respiration. Main sources of carbohydrates are starch, which is found in potatoes, and starch can be found in cereals as well, in rice, beans and peas, and in yam. As for simple sugars, like reducing sugars, you have glucose in honey, and you have sugar cane, and also fruits. Fat molecules contain more energy than carbohydrate. You saw just now that there's much more hydrogen atoms in fat, and because of that, like, they contain more bonds. That means they contain actually more energy, and these fat can be stored in the skin and around the kidney as well for insulation. The main sources of fat is in meat, dairy, egg yolk, avocado, which is a veggie fat, and nuts. Protein is used for growth and repair of your cells and tissues. The main source of protein are meat, peas and beans, fish, egg white, and peanuts. There are a few vitamins that you need to learn. So in year 8, you learn A, B, C, and D, but I'm going to only explain C and D in this video. You can check out the importance and the examples of vitamin A and B inside your textbook, and I can actually link you to the PDF version of your textbook in your Google Classroom. So what are the importance of vitamin C? It's for healthy gums and teeth. Vitamin C is also called ascorbic acid. Main sources of vitamin C are citrus fruits that are fresh and also cabbage. Vitamin D is important for the uptake of calcium. So it actually helps to take up the mineral calcium from the gut. The gut is actually a part of your small intestine. And for strong tooth and bone formation, it's also important to take up calcium, so that means that it is also important for tooth and making bones. Vitamin D is also called calciferol, just so you know. The main sources of vitamin D are fresh liver, egg yolk, and the action of the sun on the skin. So your skin actually makes vitamin D as well. Calcium is a mineral, and the importance for this is for strong, healthy bones and teeth, and for muscle action and blood clotting. What is blood clotting? Blood clotting happens when you have like a cut, for example, and then you can't have your blood drooling everywhere and dripping everywhere because you need that blood. So what happens is that your platelets in your blood is going to block that cut, the opening of that tissue, and it will form a blood clotting. So main sources of calcium are flour and milk. Iron is another mineral. Oh yeah, by the way, remember that calcium is being absorbed with the help of vitamin D just now. Okay, now we're moving on to the other mineral, which is iron. And iron is important to form hemoglobin, which is the red pigment that you can find inside red blood cells. The oxygen-carrying red pigment is important because you need to make more red blood cells in your body for respiration, for energy. The main sources of iron are liver, you have red meat, and spinach, which is a veggie. Now we're moving on to fiber. Fiber is made up of largely cellulose from the cell walls of plants. So plants actually make these fiber. Cellulose is an insoluble carbohydrate. It means that it cannot be dissolved in water-based substances. You have these things called polysaccharides, remember, which is starch, glycogen, and cellulose. So remember that starch is made up of the glucose, and then actually glucose can be converted into cellulose as well. This is the group of carbohydrate, actually fiber, which is polysaccharide. The cell wall of the plants are made up of these cellulose, and you can find this a lot in veggie and fruits. The importance of fiber is that it forms the bulk in the intestine, and it gives muscles, allowing this process called peristalsis to push against. So it helps to prevent I wouldn't call it a super disease, but it is a disease, constipation, if you have not enough of fiber in your body. So the main sources of fiber is fruit, veggies, and nuts. Lastly, water. Water is important because it acts as a solvent. So it helps to dissolve a lot of chemicals that is important in your body for chemical reactions. It also acts as a medium for all chemical reactions to occur, and it also helps you to cool your body temperature and makes up about 68% of your body mass. The main sources of water are in drinks and in all foods, even all your protein and meat. Okay, now we're moving on to, so we talked about listing out the chemical elements already. This is CHO, CHO, and CHONS. But we also need to write down step to step the test for the main food sources, which is carbohydrate, protein, and fat. And whenever you talk about the test, you also need to learn about the expected observations. So how does it look like if it's positive and a negative result? So this learning objective is to describe one test for each of them. So starch using iodine, glucose for Benedict solution, protein using the Biuret test, and fats and oils using the grease spot test, but I'm also going to explain to you this test called the ethanol emulsion test. So how do you study this? You need to write down step by step the test for carbohydrate, protein, and fat, and also all of these things, like if it's a positive test, what are the results? If it's a negative test, what are the results? So here we have food tests. Number 1, carbohydrate is using this brown color solution or chemical called iodine. Reducing sugar uses this blue, like a dark blue color called Benedict solution. Protein, we use the Biuret reagent, which is blue but kind of like a light blue color. And we have fat, which is ethanol emulsion test using ethanol, which is an alcohol, but also the one for your year 8 syllabus, which is grease spot test. When you want to test for starch, you use iodine. So what you do, the first step is to add a few drops of iodine solution directly onto the food sample, and then you observe for a color change. Mainly this is used for solid samples like that. You just drop it drop by drop. You need to also have your white tile so that you don't get to mess up your bench. And for liquid samples, you add the food sample into a test tube, and then you just add drop by drop iodine solution. This is the kind of result that you'll get if you test for starch. So what happens if it's positive is that brown color turns blue-black, and if it's a negative, then it will remain brown in color. So what are the results you're going to get? You're going to observe that there will be a color change from yellow-brown to blue-black, and the interpretation is that starch is present. But if starch is not present, the brown color of iodine will remain and no color change is observed. So you need to mention all of these things. So we talked about sugars, and there's this thing called a reducing sugar. So reducing sugars use the Benedict solution test for them to look if they're present or if they are absent. But all of these things, so you have monosaccharides and disaccharides, yeah, but not all of these are reducing sugars. You can only use Benedicts on these all of these carbohydrates. So the main one we're looking at here is glucose, but remember that sucrose doesn't react with the Benedict's test unless you add, I think, hydrochloric acid to it. Okay, so how do we test for reducing sugars, aka glucose, using Benedict's test? You place 2 cm cube of the sample into a test tube, then you add 2 cm cube of Benedict solution to the food sample, and you need to shake the mixture. So that's step 3. So you first get your white tile, you get that food sample, cut it into pieces, then you put it in a test tube. You can add a bit of water and shake if you want, but otherwise just add that Benedict solution equal volume and just shake it. After you shake it, you place the tube in a boiling water bath for 2 to 3 minutes and observe for a color change. So what you need to prepare for the water bath is a tripod stand. You add your wire gauze and some water in a beaker. You turn on your Bunsen burner and you add that mixture after you shake it up, and you wait for 2 to 3 minutes. After that, you switch off that flame and observe for a color change. So if it is a positive test, you are going to observe this kind of result. So it starts off with blue. So if there's none, it's going to see a blue. If you have a little bit of sugar, of glucose there, you will get green or yellow PPT. That is short form for precipitate. Then if you have more, you're going to get this orange color. Otherwise, you're going to get brick red, which means there is high amount of glucose inside that sample. If reducing sugar, aka glucose, is present, a blue, green, yellow, orange, brown, red, or brick red precipitate is formed. Reducing sugar is not present if the mixture remains blue in color. So this is the observation you're going to get. Test for protein using Biuret reagent. First, you need to place 2 cm cube of the sample onto a clean test tube. Then you add equal volume of Biuret reagent, which is 2 cm cube as well, and shake it. Then after that, you allow it to stand for 5 minutes and observe for a color change. So it's pretty simple. You just add Biuret reagent, equal volume, and then you shake, and then you wait for it to stand for 5 minutes. This is the kind of result you're going to get. So if it's positive, that blue color will become purple, but then if it's negative, it will just remain that pale blue color. The observation for a positive result is that the solution will change from blue to purple. Protein is present, and negative, the solution will remain blue. This means that protein is not present. For fat, you need to cut and crush the food sample, and then you place it into an empty test tube. And then after that, you add ethanol. So you have to add alcohol first when you test for fat. This is because fat dissolves in alcohol. So you put it there, and then you shake and shake and shake, and then you get your food sample. So after that, you do this thing called decanting. So decanting means that you separate the solids from the liquid mixture. Then after that, finally, you add water drop by drop and observe for change. So remember, you need to add this to a new clean test tube. So you decant it, you get only the liquidy part. And then finally, the last step, you have to add drop by drop water and then observe for change. If it is clear, that means fat is not present. But if it is like this cloudy precipitation being seen, like a layer of cloudiness, then that means that fat is present. Fat is actually very hydrophobic, so they actually do not mix well with water. You see this word lipid right here. Lipids mean fat, actually. This is the scientific term for fat. So the observation for a negative result is that solution will remain colorless. No emulsion is formed. But if you have a layer of cloudy white suspension formed at the top of a solution, so this means that there are actually tiny globules of fat, and this is called an emulsion. So food with high fat will have a higher layer of that white suspension. This means that lipids are present. Lastly, this is the one for year 8. If you test for fat using this thing called the grease spot test, this is pretty simple. All you have to do is you drop or smear the food sample on a filter paper. Then you allow it to dry. So you get that food sample and then the filter paper and just drop it or smear it onto that filter paper. And you're going to see something like this. So that is called the grease, like forming a spot on the filter paper. So if you have no translucent spot formed, so like around this area, that means that there's no lipid, no fat there. Lipids are not present. And if you have that spot like that, that means that that food sample right here actually contains lipids or fat. So translucent spot is formed. And that is the end of the video. Thank you so much, everyone. I'll see you guys next time.